Okay, so my name is Linda Guantai. I am a research associate at International Livestock Research Institute. And today I'll be talking about Kenyan Salmonella phages can reverse antibiotic resistance in field strains of Salmonella intritides. So why should we be concerned about non-typhoidal Salmonella and antimicrobial resistance? Is non-typhoidal Salmonella serovus have been shown to cause bloodstream infections in specific geographical regions, which pose a particular threat to those that are HIV or malaria infected. It is responsible for approximately 93 million enteric infections and also is associated with 155,000 deaths annually. WHO showed that the increase in antibiotic use in animals has resulted in the development of resistant Salmonella, Campylobacter, and E. coli, which contribute to human infections that are actually difficult to treat. AMR is a major concern, especially in low and middle income countries, because it is 90% of the 700,000 deaths that occur each year from AMR occur in low and middle income countries. The increase in antimicrobial usage is expected to increase by 67% by 2030, which will lead to a decrease of about 10% in livestock production in low and middle income countries. This will cost the world about $3.4 trillion a year, and 24 million people will be forced into extreme poverty. By 2050, 10 million deaths each year will be attributed from antimicrobial resistance. So some of the solutions to tackle AMR in Salmonella include probiotics, prebiotics, and bacteriophages. However, the use of bacteriophages, frequent exposure of bacteriophages to the same strains of bacteria can lead to the development of bacteriophage insensitive mutants. This is by using different mechanisms, such as using endonucleases, restriction modification, the CRISPR-Cas system, and mutation or loss of cell receptors. We can actually use the development of bacteriophage insensitive mutants to our advantage by utilizing genetic trade-offs that occur when a bacteria is exposed to both phages and antibiotics at the same time. So this is by a phenomenon which is the phage antibiotic synergy, and it was first reported back in 1945 when it was used against Staphylococcus aureus. Several receptors on the bacteria cell where antibiotics and phages attach, including the pili, porins, and efflux pumps. So for example, if a phage attaches to antibiotic efflux pumps, it can lead to the selection against these efflux pumps, which will eventually make that bacteria susceptible to the antibiotic it was resistant to. The specific mechanisms by which phage antibiotic synergy occurs are not well known, but some explanations include, if, for example, an antibiotic can cause cell filamentation, which will eventually increase phage production. And also, phages can cause bacterial extracellular membrane degradation, which enhances antibiotic internalization into the cell. Also, cell aggregation increases phage antibiotic synergy, and some antibiotics can induce prophages, which eventually lead to the cell lysis. Synergy has been shown to be working better when specific classes of antibiotics have been used, such as beta-lactams and fluoroquinolones. There are very many studies that have shown the success of combination therapy, including this study where phage and rofloxacin was used in combination, and there was a mortality rate of about 0%. And when, as compared to the phage treatment that was 15%, and enrofloxacin mortality rate was about 3%. There are also other studies which have shown that using phages and antibiotics in combination actually work more effectively in controlling biofilms. So in our study, we had collected water and fecal samples from 631 farms, from which we isolated Salmonella and Salmonella-specific phages. We obtained 16 Salmonella isolates, which were CRISPR type, and also serology and PCR was done. And we got 6 Salmonella Enteridids, which we'll be using in this study. So we also obtained 63 phages, which have been shown in a paper that we recently published. And we use one of these phage, ILRI K24, that's here, and another one, that's ILRI K23, for this study where we're showing combination therapy. So our main aim was to tell if can these phages work synergistically with antibiotics to revert antibiotic resistance. So first, we isolated bacteriophage insensitive mutants using the 6 Salmonella Enteridids I've shown previously, and ILRI K23 and ILRI K24 phages to isolate bacteriophage insensitive mutants. So we were able to isolate 44 bacteriophage insensitive mutants, and we noted that the number of bacteriophage insensitive mutants differed among the phages, with more beams being selected from ILRI when we are using phage ILRI K24. Next aim was to determine if how many of these beams could change in their AMR profile. So we carried out antibiotic susceptibility tests using 23 antibiotics. From this data, I've selected a few of these antibiotics where we could see most changes. And it was interesting to find that about 56.8% that where the host was previously resistant were now susceptible. And we had a few of these, such as this one here that is becoming resistant to nalidixic acid. So we decided to just check if, for example, there's maybe loss of AMR genes. So we decided to check if the beams lose the AMR associated gene, specifically TET-A, TET-B, TET-C, and SHRA and AAD2 genes. Some of these genes, such as TET-A, are efflux pumps related associated genes. Interestingly, we found that some of these genes that we were able to detect in the wild type Salmonella, we could no longer detect them in the beams, including TET-A in Salmonella 16, Salmonella 73, 569, and 572. 572, we could no longer detect 4 genes in the beams. So we decided to just check in general if there are any changes in the adsorption capacities of the phages. So we used the phage ILRI K24 against our exponential bacteria cultures, and determined after infection and incubation, we determined the phage titers in the supernatants. This is work in progress, but so far we've been able to see that there's actually some differences. We can see that the bacteriophage insensitive mutants demonstrate lower phage adsorption as compared to the wild type, this Salmonella 177. And also the bacteriophage, the beams, they had varied phage adsorption capacities. In summary, we observe that some beams actually lose some AMR-associated genes, and some beams have lower phage absorption capacities. So our next thing was to check what are the minimum concentrations of antibiotics that can kill the wild-type Salmonella versus the beams, and also to check how our beams, our phages and antibiotics synergize. So we carried out minimum inhibitory concentrations using e-test strips. As seen on the right is our work in the laboratory, and we could see some resistance in some antibiotics, some beams still. But generally, most of the beams here are here in black and the wild type are in red. Most of the beams were exhibiting, they required lower concentrations of the antibiotic as compared to the wild type. But some beams had developed resistance to some antibiotics such as gentamicin, as shown from this graph. We have like this beam here on top. So we combined our phage, K24, and antibiotics, specifically ciprofloxacin, streptomycin, which we carried out OD measurements at different time points, and also checked tetracycline. For this one, we did colony forming units at different time points. So interestingly, from these results, we were able to see that 5 out of 6 of the isolates there was significant bacterial reduction when streptomycin was added after 6 hours as compared to when streptomycin was added at the same time. And 4 out of these isolates were significantly reduced when ciprofloxacin and phages were combined simultaneously. It was also important to note that one of the isolates, Salmonella 569, there was no differences in either when using simultaneous and sequential treatment, because for both of them, they had significant reduction of the bacteria density. For tetracycline, we had 2 isolates that had significant bacterial density reduction when we used both tetracycline and phages at the same time. So in conclusion, 44 beams were isolated from 2 phages against 6 Salmonella enteritis, and 79.5% of the beams were actually selected with one of the phage, K24, and 3 out of 6 of the beams lost the AMR gene. Majority of the beams lost also the phage binding capacities. So in conclusion, our phages could be used as a combination therapy to control antimicrobial resistance. I'd like to thank my supervisors, Nicholas Civitek and Moses Masika from the University of Nairobi, and my colleague, Angela Makumi, Josiah Ojaba, Amos Muhoni, Ashni Mudley, and Bishon Muloi from ILRE, and also Steven Munu from University of Laval. Thank you.
So thank you, Linda. So do we have any questions from the audience?
Hello, Linda. I'm Mary from the University of Sheffield. I was wondering, do you know of any literature which shows that the combination of these different solutions basically doesn't show any effect or even has any negative results, for instance? Or is it always positive, it works no matter what?
No, actually, there are several literatures that have shown it actually depends on the antibiotic class and also the bacteria that is being used against. And there are some cases of antagonism that has occurred. So it doesn't really mean that when you combine phages and antibiotics, they have to work. But so you have to ensure that the classes of the antibiotic classes you're using are probably they can work together with the phage.
Okay, great. Thank you.
Thank you.
There was another, yeah.
Hi, I'm Dibyendu from Exeter. I wanted to ask, you said that BIMs are losing both the AMR genes as well as losing phage binding sites. So is it the same species which are doing that, or there are different subsets? One population loses one, the other, the other?
We observed, thank you for the question. We observed these from the same species, like the same isolates. Like the one that had lost that A gene was also reducing in its adsorption capacities. But I'd like to mention that this is also work in progress because we are still working on the phage adsorption capacities. But so far, it shows there is the potential.
I'm sorry, just to follow up that if you have a bacteria which loses both the AMR genes as well as the phage binding, then you can have like a other effect that it will just be no, it's fine. Okay, sorry. Yeah. Okay. Thank you.
Okay. I think that was the questions we had for you, Linda. Thank you very much for presenting here.
Thank you. Thank you for the opportunity.